Action. All right, hi guys. Welcome to our virtual classroom. Today we are going to get ready to set up our lab for corn inheritance. I got my helper right here, Alex, and my videographer holding the camera is Mrs. McGough. So she'll be helping us out as well. So by now you guys should have learned a little bit about albinism. In the videos you watched, there was a warm-up question you should have done by now, which is something I'm going to grade. So yeah, albinism is a recessive trait. It happens in lots of organisms, both animals and plants. In order for the organism to have it, it has to have both recessive alleles. So I want to kind of remind you guys about that because it's important as you start to write some of your predictions on the lab document. So there is a portion on the lab where you guys will see a Punnett square. You should be able to answer that on the Google Doc, or fill it in, I should say. So let me read to you a little bit of background, and then we're going to go ahead and talk about variables. Then we're going to go ahead and set up our experiment over here. I'm going to have Alex help out with getting all of the seeds in the soil. All right, so first of all, it says background information. You will plant, Mr. McGough and Alex, seeds today that carry genes for producing plants that make chlorophyll, the green pigment in leaves, as well as genes that lack the control of making chlorophyll. Plants have the probability of carrying genes for making chlorophyll or lacking the ability. So the lacking would be a lowercase g. Capital G would represent the ability for them to express production of chlorophyll. The genotypes of the plant seeds will be big G, so homozygous dominant, big G, heterozygous, or little g, homozygous recessive. After the seeds sprout, you guys are going to observe. I'll post another video or picture as they start to sprout for you guys to see what's happening with the seeds. And we'll have a chart where we can keep track of that. So your testable question is, how does a plant use chlorophyll, as well as what do you think might happen to a plant that lacks the ability to make chlorophyll? So kind of the cool thing is the seeds that we're using today, purchased from Carolina Biological Supply Center. They have been, these are the second generation seeds that came from two plants that were crossed. Both plants were heterozygous. So we don't know exactly what these children will be. So you guys are going to have to make some predictions. About 200 seeds were produced in this particular batch. So of those 200 seeds, we're going to see how many end up being albino and how many end up being the normal trait, which is producing chlorophyll. Okay, so we're going to set it up, but before we do that, one of the things you guys have to know, have to complete on your document are the variables, right? So independent variable, what are we testing? Dependent variable, how are we measuring it? Control variables, what are some of the things that we're doing in our lab today that are going to keep our results reliable and valid, okay? So I've taken some time and I've used my ruler to make sure my spacing is just right in my miniature cornfield of learning. All right, Alex. So here we go. I'm gonna do the first row for period 3. All right, I'm gonna plant 6 seeds. All right, about 2 centimeters deep. And then you're gonna go ahead and plant the other classes, so period 4, 5, and 7. We're gonna cover them up and we're gonna water them. So let me show you. Here are, I don't know if our cameraman, Mrs. McGough, can you zoom in? It's just a corn kernel. That's what we're planting. So I'm gonna go ahead and put the pointy end down. We got 1. They're really tiny. 2, 3. What's next, Alex? 4. Oh, which ones are going to be albino? And then the 6th one's coming up. Okay, so now Alex is going to go ahead and start planting her row just like I did. And I'll switch spots with her. So she's going to put the pointy end down. We're going to space them out. She also is using the ruler. Okay, so she'll plant those. While she's planting those, I want to tell you a couple things. Number one, some of you might want to do this experiment at home. So if you want to, you can. My recommendation is you create a flip grid, and I'll post that on our page. And in your Flipgrid, if you want me to mail you these specific seeds, you need to make a Flipgrid video where you make a position on genetically modified foods. So 2, 3, 4 minute video. How do you feel about genetically modified foods? Are you in favor of them? Are you opposed to them? Or don't you have an opinion about them at all? Okay, so first 10 people that post the Flipgrid video, I will mail you some of our Corn Inheritance seeds, our genetically modified seeds, so to speak. These are seeds that have been actually selectively bred, not genetically modified. Okay, a couple other things I want to mention to you while Alex is finishing up. We turned my front office into our classroom, so we've got our fish, our classroom fish over there, but I forget the fish's name. Is it Bob? I really forget the fish's name, so maybe someone can email me and remind me. Hope, maybe you might know. Kendall, maybe you know. Totally forget the fish's name, so maybe you can let me guys know. Anyway, so Alex has been doing a good job taking care of the fish as well as Mrs. McGough too. So when we get back, we'll put him back where he belongs. You'll notice on today's page, there's also a reassessment. So if you guys want to reassess on the genetics assessment we took before our break, you can do that. Make sure you complete the warm-up. That's 2 points. What I wanted to point out on this lab, you are going to turn in this lab twice. The first time you turn it in, it's after you complete the variables chart and the questions about predictions. So you got to complete the Punnett square. Then you have to answer these 5 questions below the Punnett square. And then you'll notice on the third page, there is a chart. And on that chart, you guys need to... Oh, Alex just said there's a worm in our soil, which is good news, right? It's going to help decompose and produce some fertilizer in there. So I guess whatever period's closest to the worm's going to have the best plants, right? Anyway, so you'll notice for a class, we'll have to put numbers. So there's 6 seeds per class. 6 times 4 is?
24.
Correct. Good job, Mrs. McGough. So you put 24 here. This stuff here, you can't do anything yet, right? So that's why it's in yellow. It says leave blank until they sprout into seedlings. But you can answer the prediction. So of the 6 seeds, how many do you predict? What do you think it's going to be? You think it's going to be 3 to 1, 3 green? I'm sorry. Of the 6 seeds, you think 3 are going to be green and 3 are going to be white? You think 4 are going to be green and 2 are going to be white? So here I want you guys to write what your prediction is and write as a ratio. How many green to how many white? Okay. You should be able to make that prediction based on what you wrote or typed in your prediction page. Once you're done filling in the chart, okay, you're going to go ahead and turn in the document. I can then grade it. Okay, and then a couple other things. We'll have a Zoom on Wednesday at 1:40 so we can check in and answer any questions you guys might have. Everything needs to be turned in this week to be graded. So you got 10 points for the variables, the questions for a prediction, and your chart. And then you have your warm-up, which is 2 points. That'll be graded for everybody. Okay, it's time to go ahead and put the soil over top.
Daddy.
Oh no. Hello, Wormy. We moved him. All right, I'll put this last one in. I can't pick it up. My hands are too. Ooh, that one's funny. It's got a hole in it. Let's not use that one. Okay. All right. So now I'm just going to take this soil and we're going to go ahead and cover up our seeds. Alex, you want to get the water right there? And just water it down the row, like where you put the seeds. You know what I'm saying? Alex is going to go ahead and water it for us. I'm going to turn the light on. Nice and hot. Here, here we go. Just go just like this. And just like this. All right. We don't need to put all of it. Just a little bit. We don't have to get it so super wet. All right, guys. Well, if you have any questions, email me. Let me know. And this virtual lab should hopefully be growing in about a week. All right. See ya.